So welcome back from the break. Hopefully you're able to grab a drink or whatever you wanted to do. If you went away or you clicked on and you're coming along with me. Let's take a look at the concept check question. How do the results of reproductive cloning and therapeutic cloning differ? Reproductive cloning, let me put it up here. Right. Reproductive cloning is geared towards producing an organism, right? It's all about reproduction. So the aim here is to produce a new organism. Therapeutic cloning is geared towards providing embryonic stem cells. Again, for therapy purposes. So the two types of cloning are aimed at producing different outcomes. One is geared towards producing a new organism. The other is geared towards producing embryonic stem cells that could be used for future therapy. So, moving along, the next topic that the book gets into, and again, you might wonder, what does this have to do with regulation of gene expression? Well, the onset of cancer has everything to do with regulation of gene expression because it's alteration in the expression of particular genes known as proto-oncogenes that takes a normal healthy cell and allows it to follow a pathway to become a precancerous cell, or a pretumorous cell, should I say, followed by a tumorous cell, and then ultimately a cancerous cell if it metastasizes and becomes a malignant tumor. This is all about gene expression. It's all about changing the output of genes that under normal circumstances have a perfectly good role in controlling cell division. It's when those genes get altered and the role of those proteins changes so that they no longer regulate cell division that we get into the issues associated with cancer. So I don't really like the way this is phrased. A gene that causes cancer is called an oncogene. And the reason why I don't like that, the way that's framed, even though this comes straight from the textbook, that's how it's framed in the textbook, is because there is no gene in your genome whose role is to make you sick. There's no gene in your genome whose role it is to give you cancer. Every gene that you possess has a normal, healthy functioning role. It's when the gene gets altered, when it gets mutated, that it then changes its role. So just bear that in mind. There is no such thing as a cancer-causing gene. There are genes whose role is to regulate cell division. When they get mutated, then their role changes. They no longer carry out their normal function, which leads to the development of cancer. But these genes do not cause cancer. That's not their job. It's when they get mutated that they allow the development of cancer because they're no longer carrying out their normal job. So not surprisingly, many of these genes that are known as proto-oncogenes are growth factors that are involved in either allowing guiding the cell through the cell cycle. We talked about that in mitosis, or send signals to the cells that it's appropriate to grow and divide. They are external factors that trigger the changes internally that lead to gene expression that allows the cell to grow and divide. There's the connection to regulation of gene expression. This is why talking about the onset of cancer, the development of cancer cells, is connected to regulation of gene expression, because it's the change in gene expression that leads cells to becoming cancerous or precancerous. So here's a way in which a gene could become an oncogene, go from proto-oncogene to oncogene. So you simply get a mutation that changes it from its normal activity, which in this case is stimulating cell division, to a protein that overstimulates cell division. And if it overstimulates cell division, you're going to have a cell that is growing and dividing quite rapidly, whereas it shouldn't be. Or if this gene gets replicated, so there are multiple copies of the gene, now you're going to have a lot more stimulation, because you're going to have a lot more of this stimulating protein, this division stimulating protein present in the cell. So it's going to be more active. Or you have a single copy of the gene and you get a mutation in the regulator region of the gene, and that changes the expression of the gene so that it gets overexpressed and you get far more protein than you ought to. And again, it's going to overstimulate the cell so that it's going to overgrow and overdivide. Any of these three changes could lead to a cell that is no longer a normal healthy cell, but it now would be considered to be pre-tumorous, or tumorous, or precancerous, or cancerous, depending on how many changes have occurred. Because cancer is never the product of a single gene change. There are always multiple gene changes associated with the onset of cancer, as you'll see. Now, one set of genes that routinely get altered during the development of cells from normal healthy cells to cancer cells are a set of genes known as tumor suppressor genes. And their name tells you pretty much what their job is. Their job is to regulate cell division and to stop cells from growing out of control, from basically infinitely growing and dividing. And when you get a mutation to one of the tumor suppressor genes, the most famous of which is known as P56. P56 is a tumor suppressor gene that is supposed to cause cells to die, undergo a process called apoptosis, when they become precancerous or pretumorous, when their DNA gets damaged sufficiently that they should not be allowed to divide, or you get an accumulation of mutations that means that they are on the path to becoming tumor cells. P56 is supposed to stop them from doing that by pushing them into what you might call programmed cell death or programmed cell suicide, otherwise known as apoptosis. But when P56 or these other tumor suppressor genes get mutated, they lose their ability to regulate cell division, and that allows the cell to start growing and dividing in an uncontrolled fashion. So tumor suppressor genes are almost always altered during the onset of every type of cancer, because these are the genes that are supposed to prevent that from happening. So it stands to reason that when cancer starts to develop, one or more of these genes should be mutated to allow the genes to start, the cells to start growing in an unregulated, uncontrolled fashion. So this shows you basically what I've been talking about. Here's the tumor suppressor gene doing its normal job, regulating cell growth so that it is normal growth as needed. When it gets mutated, however, it no longer is able to regulate cell growth, and the cells start growing in an unregulated fashion. You have the onset of cancer, or at least the development of cells in the direction of cancer. So I said right back at the beginning that I would talk about the use of avatars to determine effective cancer treatments. Remember, different types of cancer respond to different combinations of drugs. Even two cancers of the same type may respond to different type combinations of drugs, because every person's version of a cancer is unique to that person. That's what it's turning out to be. So one of the ways that you can get around this issue of different versions of the same cancer responding to different types of drugs is to actually test it out in a mouse model. And there have been mice developed that lack an immune system. These are hairless mice. They've been created specifically to lack an immune system, which means that when you add human cancer cells under the skin of these mice, those cancer cells begin to grow. And so once you have the tumors growing on the backs of these mice, you can then give the mice the different combinations of drugs that you want to use or you suspect might be useful against a particular type of cancer that you've placed on this mouse. So here's basically the flow through the mouse avatar system. You take the tumor cells out of the patient, insert the tumor cells underneath the skin of these hairless mice that have no immune system, and you treat each of the mice with a anti-cancer drug, and you monitor the mice to see which of the drugs is most effective against the particular cancer, which of the particular drugs is effective against this cancer. That's the drug then that you would start using for the patient. You might even use some of these drugs in combination depending on the type of cancer. So in the particular study that's referenced in the textbook, 14 patients had tumor cells taken from them and placed on the mouse avatars. Of the 14 patients, unfortunately, 2 patients did not respond, or the tumors in the mice did not respond to any of the treatments. So there was nothing that could be done for those 2 patients, based on this study at least. Of the remaining 12 patients, 1 patient died before the avatar study was completed. Of the remaining 11 patients that whose avatars completed the process, they had a treatment that was discovered through the avatar system that was effective, either in the case of a single drug or combinations of drugs that were shown in the avatar system to be effective against the cancer. What this is indicating is that an individualized cancer treatment, a cancer treatment that's tailored specifically to you and your version of the cancer that you're suffering from, can be, and most likely will be, the most effective way of treating your cancer. So there are all sorts of ways in which we can personalize these. This is one way, using this avatar system. But probably a more effective way of doing this is genome sequencing, whole genome sequencing, and looking for the mutations that gave rise to your cancer as a consequence of screening your genome, and then applying a cancer treatment or a combination of cancer treatments based on the treatments that are known to be most effective against the mutations that your cancer happens to possess. That's where individualized medicine is heading at this point as far as cancer treatments are concerned. So the next couple of slides, or at least the next slide, just simply illustrates the idea that multiple changes are necessary to give rise to cancer. Colon cancer is one of those cancers which ought to be caught relatively easily, because it's fairly easy to recognize when things are going wrong in the colon. If you look at your fecal matter and you notice that there is fresh blood in it, that could be an indication that there's something going on in the colon. That's evidence of bleeding. And bleeding in the colon is one of the early signs of the presence of colon cancer, while it's still localized inside the colon. You could also get a colonoscopy. The rule used to be once you reached 50, you would have a baseline colonoscopy, and then depending on what was found, you would have follow-ups on a regular basis. Now the recommendation is 45 years for the baseline. because we are unfortunately finding colon cancer in younger and younger individuals. So they're starting the use of baseline colonoscopy at younger ages to catch more of the colon cancers. And of course, if you have a colonoscopy, if something is found like a precancerous mass, they can immediately remove those. Because once you get the polyp out, which is how it first appears in the colon, there's likely no further treatment required because you've removed the source of the pre-tumorous mass, the precancerous mass. And what they'll typically do is do another colonoscopy fairly quickly, probably within 1 year, just to check and see if anything has changed in that 1 year, because colon cancers grow fairly quickly, unfortunately. And if they see nothing, then they'll put you on a more relaxed procedure where you have one every 3 to 5 years until they discover something again. So what sort of changes are required? Well, part of the problem for this with colon cancer is that there are lots of little blood vessels associated with the colon in order to transport water and other material away from the digestive tract. So when the cancer cells first start to emerge in the colon wall, at this point, they're not particularly dangerous. When they develop into the polyp, which is a benign tumor, they're still not particularly dangerous. And if they get caught in a colonoscopy, the physician doing the colonoscopy can remove the whole polyp. That will be sent away for testing by a pathologist, and they'll make sure that they've got the whole polyp out. And then you'll get a follow-up colonoscopy just to make sure that nothing's happened. If it develops into a malignant mass, however, it grows into the wall of the colon, that allows cells to enter the bloodstream and then move away to another location where a new tumor, which now will be a malignant tumor, and therefore you officially have cancer, will take place. You notice that it is multiple changes that are involved here. This is showing 3 changes. My understanding is that it's a little bit more than that. It's around 5 or 6 changes that are necessary to develop from normal healthy cells through pre-tumorous cells to the benign polyp through to the malignant tumor. There are about 5, maybe 6 changes that are required in total. But it means that multiple changes need to occur in order for this to happen. So when a pre-tumorous mass develops into a benign tumor and then develops on into a malignant tumor, it's all about changes of proto-oncogenes to oncogenes and then loss of tumor suppressor genes, which we've already talked about. It's those changes, it's the multiple changes that lead to the development of malignant tumors. So here is again a diagram that suggests what the changes could be like. This is just a suggestion. This is not saying that it is an actual pathway to a cancer. It just shows that it could be different genes in different locations on different chromosomes that need to be altered, but ultimately it's the combination of all of these mutations that take you from a normal healthy cell to a pre-tumorous cell to a benign tumor to a precancerous cell to a malignant cancer cell. Multiple changes are required. Of course, if you have cancers, a history of cancer in your family, that tells you that some of these changes are being inherited from one generation to the next. Some of these changes are already present in the DNA that is being passed on from one generation to the next. And that simply means you have a higher chance of developing cancer because some of the changes that need to accumulate have already occurred and you've inherited them in that fashion. This is the reason why we stress the necessity for early detection. The earlier you can find the onset of tumors, particularly if they are benign, the greater is your chance of a potential cure. You saw the statistics for breast cancer at the beginning of the presentation. That's pretty common amongst most cancers. The earlier you can get them, the more easily it is to treat them with this surgery without having to resort to radiation and chemotherapy because you take out the benign tumor, the greater is your chance of survival and cure. Remember, 5 years, the magic time. If you can stay free of the cancer for 5 years, you're considered to be cured. Doesn't mean you won't get it again. It means that in this particular instance, you're cured of that cancer. Early detection is the key. So this just gives you a little bit more data about some of the cancers. So in the case of colon cancers, about 15% of colon cancers are or have an inherited component, let's put it that way. In the case of breast cancers, about 5 to 10% of breast cancers are associated with these two genes, the BRCA1 and BRCA2 genes. Just bear in mind, however, that it's not just these genes that are responsible for breast cancer. These are, by the way, BRCA genes are also associated with ovarian cancer, cervical cancer, and for men, testicular cancer. An association with testicular cancer was identified about 2 years ago in the case of the BRCA genes. And guys, you should also remember that you have breast tissue, so the BRCA genes could give you breast cancer. You have breast tissue, it just doesn't develop because it's not exposed to estrogen and estradiol as it would be in a woman. So you still have the breast cancer, you're still capable of getting breast cancer. So you have breast tissue, you can get breast cancer if you are BRCA positive. Some people have taken an extreme approach to finding that they have the BRCA genes. So the most famous individual here would be Angelina Jolie. She was discovered to be BRCA positive. And so she had a radical mastectomy of both breasts and also had her ovaries removed. On the basis that if you take out the tissue that is likely to develop into cancerous tissue, you remove the possibility of developing cancers associated with being BRCA positive. Whether that is a feasible approach is a personal decision. Different people take different approaches based on their personal preference. So a quick break before we get into the last section, I believe, of the chapter. How can a mutation in a tumor suppressor gene contribute to the development of cancer? Well, that's something that we've just been talking about. Remember, the idea of a tumor suppressor gene is that it regulates cell division. So suppressor genes regulate cell division. Loss of these genes leads to unregulated cell growth and division, which by definition is cancer. So that's how a mutation in a tumor suppressor gene could lead to the development or could contribute to the development of cancer. You lose the regulation of cell division as a consequence of the mutation. So one of the unfortunate things about cancer is that it is a leading cause of death. As much as we put emphasis on early detection, on lifestyle, paying attention to what your body is telling you, avoiding contact with chemicals that are known to be cancer-causing agents, or situations like exposure to ultraviolet that can increase the chance of mutations in the skin, for instance. The hard reality is cancer is a disease of age, and the longer we live, the more likely it is that we will develop cancer. In fact, the numbers are pretty scary when you think about it. It is generally considered that about 1 in 3 individuals will have some interaction with cancer throughout the course of their lifetime, just because we live as long as we do these days. Some people will obviously have their encounter much earlier. Some people will have their encounter much later. Some people probably won't have any encounter at all. It is a 1 in 3 chance. But there are certain things that we know, if you do them, will increase your likelihood of developing cancer. So tobacco is the leading cause of cancer, by far the leading cause of cancer. And there is no safe way of using tobacco. It's as simple as that. There is no safe way of consuming tobacco. Whether you smoke it, chew it, snort it up your nose, which some people do, it's called snuff. Very popular back in the 17th and 18th century in Europe amongst the gentry. Some people still do it. There is no safe way of using tobacco, because there are chemicals in tobacco that are known carcinogens, and your exposure to them simply increases your risk of cancer. We know that ultraviolet light, in whatever form you get exposed to it, whether it be natural sunlight or a tanning bed, can lead to mutations in the DNA of your skin, which leads to skin cancer. And skin cancers, particularly melanomas, are some of the fastest growing cancers that we know of, and therefore are some of the most lethal that we know of. Because again, as we saw in the colon, where there are the blood vessels that the cancers can get into and move to other locations, so underneath the skin, under the skin bed where your new skin cells come from, are lots of tiny little capillaries. So when the melanoma grows down into that, it can very easily move to a new location. Something that I did not know until a few years ago is that you do not need to get exposed to ultraviolet radiation to develop a melanoma, because you can actually get melanomas of internal tissue. Yeah, I actually met someone who was fighting a melanoma of internal tissue, and unfortunately she subsequently passed away about a year or so ago. But she fought for 3 or 4 years against this cancer, going to the Mayo Clinic and getting involved in some of the cutting edge treatments for cancer that helped her for a while, but eventually the cancer just grew out of control, and it wound up killing her about 18 months ago. So yeah, melanomas are pretty bad news. But obviously there are things that we can do to prevent these sorts of cancers. Colon cancers, we can do the testing. We should be checking tissue that we know is susceptible, and we should also be avoiding the things that we know can increase the likelihood of cancer. And yet people still do the things that we know are going to cause cancer. I'm not sure why, but they do. There are some things that we can do as far as diet is concerned. This one about plant fiber, that one I know goes backwards and forwards about whether this is advantageous or not. It's specifically geared towards colon cancer. Every so often, I read something that says, yes, it's effective in reducing the chance of colon cancer, and then a few years later, someone says, no, not so much. So this one I'm not terribly sure about. Eating less animal fat, most definitely. Saturated fats, because of the way they are processed in our body, can increase the likelihood of some cancers. Vitamins C and E are both antioxidant vitamins, and antioxidant vitamins are known to reduce the chance of damage to the DNA. Because when you have oxygen-free radicals, which is what these vitamins protect you against, loose in your cells, they can damage the DNA. They cause random damage in the DNA. If it happens to be in the wrong spot in the DNA, it can lead to a change that could change a proto-oncogene into an oncogene. Then the cruciferous vegetables, which are these ones. So cruciferous vegetables. So it's broccoli, cauliflower, Brussels sprouts, kale, bok choy, cabbage, all of those relatives, all of those plants, they are related to each other. They are derived from a common ancestor, a little mustard plant that grows along the shores of Europe and Asia. And it was selectively bred by people to favor particular traits. So when you think about cabbage and broccoli and cauliflower and kale and kohlrabi, Brussels sprouts and so forth, they all belong to this family of cruciferous vegetables. They have sulfur-containing compounds in them. And these sulfur-containing compounds have been shown to be really potent anti-cancer chemicals. This is the reason why eating some of these on a regular basis is a good thing as part of your diet. Because we know diet has a very distinctive role in the onset of cancer as well. It's not just your family history, it's not just what you do during your life, it's also the things that you place in your body, the food that you consume, and other things that you consume. So this diagram shows the correlation between different types of risk factors and their contribution to cancers in terms of the number of diagnoses and the number of deaths, using the latest figures that we have available to us, which are the 2017 figures from the American Cancer Society. When you look at these figures, there's a checkpoint question here, and I know I've got this checkpoint question at the end of the presentation, but let's do it here. When we look at these statistics, and we look at this question of all the known behavioral factors, which one causes the most cancer cases and deaths? It's pretty obvious, right? It's got to be tobacco. It's there, it's there, it's there, it's there, it's there, it's there. Tobacco has a role in each one of those cancers, right? Consumption of tobacco in some form, either smoking, chewing, snorting, however you use it, has a role in all of these types of cancers. It's not just lung cancer. It's multiple cancers that we now know use of tobacco plays a role in. I'll just let that sit. You can make of that as you wish. So, yeah, this slide and the next one is actually getting to something that I just recently read about in a book on cancer that I have in my library. It's a relatively new book on cancer. One of the most interesting ways that oncologists, the people who treat cancer and study cancer, are starting to look at cancer is by applying our knowledge of evolution. And you might think, how does that work? Well, remember that natural selection, as described by Darwin and Wallace, tells you how organisms can evolve and change over time. And when you think about cancer cells, that's what they do. They start out as normal, healthy cells, and they evolve over time into tumor cells and cancer cells. And so if we apply some of the ideas that Darwin and Wallace proposed as supporting assumptions for the process of natural selection, then we start to get a new vision of what cancer is all about. So one of the issues, or one of the proposals, or one of the observations, I guess, that Darwin and Wallace made is that every population produces way more offspring than the environment can support. And basically, that's what cancer cells do. They overproduce themselves. They grow in an uncontrolled fashion. So they are clearly producing far more cells than they ought to be producing. They're overpopulated. Then a second idea is that there's supposed to be genetic variation amongst the individuals, which leads to phenotypic variation or physical variation, if you forget what phenotypic variation means. Hopefully you remember. And we see that sort of variation in cells within a single tumor. Two cells adjacent to each other in the same tumor can actually have different sets of genetic changes, which is kind of scary, because what that says is if you treat a tumor with a single drug that works against some of the genetic changes that have been identified, you'll kill off some of the tumor cells, but you may not kill off all of them, because those that have mutations that simply mean they're less susceptible to the cancer drug are going to survive. Just as bacteria become insensitive to antibiotics, so tumor cells can become insensitive to cancer drugs. Let that sit for a moment. This is something that oncologists have only just really started to understand in the last 10 years, that not all cancers are the same. Two individuals that have the same cancer can have totally different mutations, or a different combination of mutations that led to that exact same cancer. And even within the cancer, there can be cells that have different accumulations of mutations. And it's only because of the ability to be able to sequence genomes and to be able to amplify DNA, something that we'll be talking about in the last chapter of the semester in our next presentation, when we talk about DNA technology. It's only with the availability of that technology that oncologists have been able to divide tumors up into smaller and smaller clusters of cells and almost be able to look at individual cells within a tumor and be able to look at the mutations that each of those individual cells have and whether they have the same set of mutations. And this is where we've started to understand that not all cells within the same cancer are even identical. And of course, those cells that survive the initial treatments are the ones that are more likely to produce offspring that are more likely to develop into malignant tumors. In other words, cancers can very distinctly evolve. Just as populations of organisms can evolve, so populations of cancer cells can evolve. Which means we need to start thinking about cancer in a totally different way. And we need to start approaching the treatment of cancer in new and different ways. Because we can no longer assume that a single cancer can be treated with a single treatment, or that the cancers of two individuals can be treated the same way. Because that may not necessarily be the case. It almost certainly is not the case. But now that we start to understand these things, it also opens the door to the development of lots of different ways of treating cancer and for personalizing the treatment, which in the end is likely to give us a higher likelihood of survival if the cancer treatment can be tailored specifically to our type of cancer and our particular version of that type of cancer. That holds out the hope that not only do we have to think about cancer in a different way, but in developing these personalized treatments for cancer, that we have a greater chance of providing a cure for an individual's cancer. Kind of an exciting idea. So that's the end of this chapter on gene regulation. Some of the topics initially did not seem like they involved regulation of gene expression. But as you start to dig deeper into them, you start to realize that, in fact, they really are all about regulation of gene expression. Whether it be regulating the production of proteins as a normal function of cells, as cells grow and develop, in regulating the expression of genes in stem cells so that you can de-differentiate them or redifferentiate them or take them in different directions to become different types of cells to be used for therapy, or for cloning whole organisms, or for treating cancers in very specialized, novel ways that are tailored to specific individuals. It all comes down to understanding how genes are expressed and what are the mechanisms that are involved in regulating the expression of genes in different types of cells. It really does come down to understanding how genes are regulated and what the consequences of that regulation are for the expression of those genes in cells, and what the consequences of those expressions in the cells are for the development of the cells. What do they become? And what consequences can that have for individuals? So that brings us to the end of this chapter. We've already looked at this concept check question. So we don't have to worry about it again. So as always, if you have any comments, questions, or concerns about anything that I've covered in this chapter, by all means, come and check with me in Office House, and I'll be happy to help you out. Otherwise, I'll see you back here one more time for Chapter 12, which is looking at DNA technology, and there's some really cool stuff in there. I'll see you again.